I would like to welcome Professor Magdalena Götz from Germany to give the Heller lecture today. Magdalena is the director of the Institute of Stem Cell Research Helmholtz Center in Munich since 2004. She's the chair, since 2011, she's also the chair of the Physiological Genomics Biomedical Center at the Ludwig Maximilian University of Munich. And I don't have time to go through the 25-page long CV that I got, but just to give you a taste. There's a long list of awards and honors, to name just a few from the past few years. The Bavarian Constitutional Medal, the Betty and David Koster Foundation Brain Research Award, the Jacob Hertz Prize, the Mendel Medal, the Schellenberg Research Prize, Roger, the Spielberg Prize, and this list goes on and on and on. Magdalena sits on numerous editorial boards of journals such as Cell, Cell Stem Cell, Neuron, Embryo Journal, Genes and Development, and this list goes on and on and on. Magdalena also sits on numerous grant panels, organizational boards, societies, conferences, advanced schools, and by this contributing to the community as a whole. Magdalena also has a long list of trainees that she has mentored. These trainees are now prominent professors themselves in numerous places around the globe and in some of the best universities. And Magdalena, of course, has authored many publications, in fact over 200 publications, that made huge impact on our understanding of the molecular biology of brain development. Like, for example, discovering that glial cells are the neural stem cells of the developing brain. And she also made major contribution to diverse fields in developmental neuroscience, such as adult neurogenesis, reprogramming, neural regenerations, and more. So you are indeed, Magdalena, an inspiring scientist to all of us, and for human scientists especially. And in this context, I would like to mention to the women scientists among us, the students, the event that Imbal is organizing after the session to speak with Magdalena. So thank you very much for the invitation. Just a final word about the Heller Lecture Series. So you were invited as part of the Heller Lecture Series in Computational Neuroscience. This series is a result of the generous support from Sir Michael Heller and Lady Morven Heller from London, which are good friends of the Hebrew University and of Israel as a whole. Sadly, Michael Heller passed away recently. Morven, however, has decided to continue to support this endowed lecture series and continue to help us at Hebrew University to welcome prominent scientists from around the world like Magdalena. I do need to do this as part of the ceremony, so I would like Magdalena to award you this certificate of recognition and appreciation for your contribution to academic inquiry and exchange at the Edmond and Lily Safa Center for Brain Sciences. And this is signed by myself and Professor Jonathan Levinstein, who is the director of our PhD program.
Thank you. I'm very honored to give this lecture.
Good. So we're going to talk about this study.
Okay, well, thanks a lot, Adi, for all your hospitality and the wonderful day yesterday. So I had a great time already. Also this morning, hearing about the exciting science going on here. And for me, it's always special to come. And my first special visit was actually at the beginning of 80s, and I told to some of the students and postdocs that in fact I came to Jerusalem to have my first lab experience at that time in Hadassah Medical School. So this was always very influential. And of course, I also feel very moved to be here at this very special moment in time in your history, where you have to stand up and fight for democracy. And this is a very important moment, and I would like to give you all support from my heart for that. And before I start crying, I better talk about science. So stimulated by this organ, and I guess this is what we share amongst all of us here. And this organ endows us with the capacities that we enjoy so much and that make me talk and you understand. But of course, not only us, but many species are equipped to cope with their local niche that they live in by the brain. Now, the brain changes in phylogeny, but changes in phylogeny and evolution are achieved in development. This is why development can shape these different brains, and for example, for electric fish, enlarge the cerebellum so much, and for us mammals, enlarge the dorsal telencephalon so much. Now, how are such changes brought about in development? In a very simple way, this brings us to the cells generating the brain. These are the neural stem cells, and here I introduce you already to the region that I will talk about a lot, the cerebral cortex, the dorsal telencephalon, and this is a view of the mouse brain at the time of neurogenesis, and the neural stem cells are here in green. So in order to enlarge a brain region, what you do is you amplify your neural stem cells, you amplify the neural stem cells, and you make them to generate more neural stem cells. This is how you enlarge a brain region, because this is like an epithelial sheet, literally. Now, of course, this will not help much if you have lots of neural stem cells, but no neurons and glia cells in your brain. So the other thing you need in order to expand the number of neurons and glia cells in a given brain region, now here the cerebral cortex, is to generate lots of neurons and glia cells, and for this you need the differentiating neural stem cells that generate an intermediate set of progenitors. Let's say we call them the transit amplifying progenitors in our field, but just see that as an amplifying machinery of cells that can then increase the neuronal and glial output. So this is, of course, a fascinating process in itself, so we think at least, and I know I share this interest with some colleagues here, but of course it has further repercussions, and really for us it was these repercussions that brought us to brain injury and repair. Because when we study neurogenesis, when and where it works best in development, and when it still works in the few adult niches where it continues as adult neurogenesis, and again we have some experts here, then of course we study the principles of neurogenesis, and this may help us to elicit neurogenesis when it's needed, namely after brain injury. Now, we could have been clever and thought about this before, but we didn't, which may mean we weren't clever or aren't clever, but the thought to do so was really triggered by our first and then Arnold Krikstein's and Takaki Miyata's finding, as mentioned by Adi, that in fact the radial glia cell, so a type of glia, is actually the neural stem cells. To put it very bluntly, if these types of glia cells, and indeed the adult neural stem cells, were identified by Arturo Alvarez Furia as also as glial in nature. So if these two types of glial cells can generate neurons, maybe we can learn something from these glial cells to instruct these glial cells, the reactive glia in an adult mammalian brain, to generate neurons. Because, as you know, after a brain injury, for example a stroke, we lose neurons, we also lose some glial cells, and they are not replaced, the neurons. And the loss of function, of course, relates to the loss of these cells. So basically, if we want to improve this situation, we have to make more neurons, and we have to deal with the scar formation. But I use this slide to illustrate that. So basically we have two tasks. Reduce the scar formation after an injury in the brain, because like any scar, even in the skin, a scar is a non-functional tissue. Likewise in the brain, the scar region is not functional. So reducing the scar formation will bring back part of the function, but then of course we need some neurons also to execute these functions. And as you know, the main avenue so far that is also already in the clinics is to put in new neurons by transplantation. We started an approach called direct neuronal reprogramming, where we are trying, and I show you where we are with this, to tackle both of these issues at the same time, namely to convert directly these reactive glial cells into new neurons. And I will show you and tell you how we did this. Now, within these questions, we came across a new principle, or a principle that maybe we discovered newly, that I find very fascinating, and I'd like to discuss this with you today as the red thread of this talk, namely the principle of organellar heterogeneity. And I'll discuss it first in development, and then I'll come to neuronal repair, replacement by using neuronal reprogramming, and I continue with this theme, but also show you where we are with this. So, what does she mean by organellar heterogeneity? Now, you all know about organs as compartments in the cells that have certain functions, like the centrosomes, the little dots here. They orchestrate the cytoskeleton. The mitochondria, here green, they are like the powerhouse of the cells. And in the nucleus, of course, we have also many compartments, but here outlined are the nucleoli, and the nucleoli are important in translation, for example. So, we all know these organelles are there. Every cell has them, every eukaryotic cell. So the assumption was, and they perform the same functions, so the assumption was they must also be very similar. What I will show you is that this is not the case, but rather that these common organelles exert their function in a highly specific manner, and this matters. So I will show you that centrosomes between very closely related cells even differ by more than half of their composition. I will show you a similar theme for the mitochondria, and I will show it to you for one protein for the nucleoli. And I will show you that this matters, otherwise I wouldn't bore you with that, for disease and neurogenesis and for direct neuronal reprogramming. So this is why I think this principle is important, and I'd like to discuss it with you then. And of course I'd like to convince you of what I'm summarizing already for you here. Now, I've already introduced you to the self-renewing neural stem cells that enlarge a brain region, and the differentiating neural stem cells that generate all the output. And of course, we could learn from this. There's this cold wind blowing at me, so I try to stand here. And of course, we try to understand molecularly what makes them different, what makes a differentiating stem cell different from a self-renewing neural stem cell. So we profiled them by isolating them by fluorescent activated FACS sorting, and over the years could identify some interesting players in that. And I'd like to start with this more recent one, the 80 hook transcription factor AGNA. So that we found to be enriched in this population of neural stem cells that generates these, let's say, the output of the neural stem cells. Now, it was in the database as 80-hook transcription factor. It turned out, as Hermann found out, that it neither has an 80-hook, nor is it a transcription factor, but in fact, it's a centrosomal protein. And I like to include this example also for that reason. Never believe what is in a database. Better check it yourself. So, Hermann raised specific antibodies against ACNA, was staining the cells, the neural stem cells, where we isolated it with this antibody, and found that it's not in the nucleus where transcription factor should be, but it's at the centrosome. We confirmed that. This is published by many means. And of course, we also looked at its function, and I'd just like to show you one example for that. When we overexpress ACNA, so it forms little hubs of ACNA protein, you can see that these are sufficient to organize microtubules in a microtubule organizing center. Now, this then led us to a new mechanism of how these differentiating neural stem cells actually delaminate from their niche, from the stem cell zone, by recruiting proteins, these red proteins, that weakens the junctions between the cells, and then by organizing the microtubules at the centrosome so the cells can move away. And this function was not specific for neural stem cells, but in fact it plays a general role in other cells like mammary epithelial cells and other epithelial cells, which may also implicate its role in metastasis formation. Now, the real interesting finding in this study for me then was the high specificity by which we found this protein at the centrosome. Because as I already told you, we found it only in subtypes of neural stem cells and not those that cell for you. And even during the neuronal migration, it's basically at the centrosome, and then again it needs to be downregulated. High specificity. So that, of course, made me think about these centrosomes, and I was wondering if ACNA is a very special protein, so it may be regulated so specifically. So I wanted to look that up. How do the centrosomes differ of neurons from, let's say, other cells, or neural stem cells from other cells? But it turned out there was no centrosome proteome of any cells from the brain, probably due to the concept that centrosomes are anyway similar between the cells, so why do we need it from different cell types? We have the great HeLa cells or HEC cells, so that's all good. Now, we were wondering, of course, if in fact we have seen only the tip of icebergs, and in fact there may be many more differences between centrosomes than anticipated. So that's what we did. We looked into centrosome heterogeneity. Now, we used a very simple 2D model of IPS differentiated into neural stem cells and neurons, and we did that because we can have these cells pure. So as beautiful as some 3D models are, we needed for that biochemical analysis pure neural stem cells without contamination of the amplifying progenitors or neurons at day 15, this is the case, and then pure neurons without any contamination of stem cells or progenitors. This is what we got, and then what first Adam, a fantastic postdoc, and then Fatma, Adam for the neural stem cells, Fatma for the neurons, IP'd 10 centrosome proteins. Now the good thing about this spatial proteomics, as this is called, is that you can actually choose as base proteins that are at different positions of the centrosome, and this means then that when you look at your reproducible interactors that you pull down, you also know where these interactors are actually localized. Now, first the answer to the first question, did we find any new proteins? Yes, we found a couple of 100 proteins that were not in any of the known centrosome protein databases. What about the neural stem cells and the neuron comparison? We found that 50% of all centrosome proteins differ between neural stem cells and the neurons. That means that during neuronal differentiation, this pan-cellular organelle, the centrosome, is basically refurbished by half of it. Now, as I mentioned, this spatial proteomics allows us to see with which baits these proteins are interacting. And as you can see, many orange dots here, these are the proteins interacting with these baits, specifically only in the neural stem cells. And these are bait proteins that are localized at the mother centriole and these appendages where cytoskeleton is organized, to put it very simple, while most of the neuron-specific proteins are actually interactors of these SEP63 proteins, only in neurons, not in neural stem cells, and we don't yet know what this means, and we can discuss more about it. Now, we were very fascinated by the fact that we also found not only individual proteins changing, but entire functional terms. Because when you plot the functional terms of all the centrosome database, you get, of course, mitotic cell cycle or cilia formation, known centrosome function. But the top geoterms in our analysis relates to RNA. So what do RNA binding proteins do at the centrosome? I will show you at least one example for what we think they do. But just to highlight in this slightly mutated slide here, that also some of the well-known players for development, like Fragile X proteins, are localized at the centrosome, and that again, these RNA-binding proteins, they differ not only in identity, but also position at the centrosome between neurons and neural stem cells. Now, obviously, I wouldn't bore you with that if it wouldn't matter, and I told you it matters for development and disease. Development, I will show you in a second, and disease, I show you here. Because we were simply wondering if this new resource, if you want, this centrosome proteome, could help us prioritizing any gene mutations that have not yet been associated with a disease because they are gene variants or basically in a single family. So we overlaid therefore the known centrosome data set and ours from neural stem cells or neurons with neurodevelopmental diseases, autism spectrum disorders, highly significant overlap, but with all centrosome databases because in fact proliferation is affected in this disease. We overlapped it with many other diseases, and I'd like to focus where we found more specific significances turning up, and I'd like to focus on this disease, the periventricular heterotopia, because as you can see, there is no significant overlap with all the known centrosome database, but only with the neural stem cell centrosome database. So somehow there must be something important about mutations of proteins that are at the centrosome only in neural stem cells. So, of course, before we get excited about this overlap, let's probe it for a single candidate, and we choose a candidate from our top geoterm, namely RNA interaction, and we choose a candidate that is expressed in all cells of our body because it's a splicing factor, PRPF6, mRNA processing factor 6. It's a ubiquitous protein, also in development, but when mutated, it causes a disease that was known already in the retina, in retinitis pigmentosa, with this mutation, and the gene variant that we found to be selectively enriched so to say, in this overlap, is this gene variant in one of the RNA binding domains here. So could it be that a CNS phenotype is brought about by the localization of this protein at the centrosome only in the neural stem cells, but not in other cells like HEC cells, for example, as we have shown, and I'm not showing you all the data. So Adam then simply models this mutation in the development of the cortex. As many of you know here, you can do so by simply electroporating the neural stem cells that are located at the ventricle by injecting the DNA and then basically pushing it by electric pulses into the tissue. By that, you can then come back some days later and look at all the expressing cells. Typically, you use a GFP to monitor these cells. And of course, they migrate, stem cells generate the transit-amplifying progenitors and the migrating neurons, and you can see how they distribute. When we overexpress the wild-type version of this splicing factor that we find also at the centrosome, you can see that we get an increase in neuronal differentiation, so to say, more cells in the cortical layers. But when we use the mutant form, indeed we get significantly more neurons at the ventricle. Just like it's the case in the periventricular heterotopia, I think the name says everything, in the patients. Interestingly, the mutation that was identified in retinitis pigmentosa did not exert this effect. So then, of course, we wanted to know then what does PRPF6 do in the developing cortex. So we sequenced the cells that express the mutant PRPF6, and we found that indeed there is changes in splicing. We took one target that was misspliced, that is in fact a kinase that phosphorylates microtubule-associated proteins. And I've shown you before that the microtubule skeleton is important for the delamination from the ventricular zone. And indeed, we could then show that we rescue the phenotype by expressing the correctly spliced form of this enzyme, but not the incorrectly spliced. And therefore, this led us to the model that, in fact, PRPF6 at the centrosome may exert a role in migration, because remember, when it's mutated, the cells get stuck at the ventricle. And by bringing the RNA, and here you see the RNA of this enzyme, to the centrosome for local translation and exerting its function there. This is partial evidence, of course. But maybe if we zoom out again, what is important is that a ubiquitous protein, when mutated, exerts a phenotype in a specific organ, at least correlated to its location at a specific organelle. And so this is maybe the main important message of why it's important to know which proteins are at the centrosome, and bear in mind that they differ so much. So I hope with this I could convince you that cell type specific centrosome composition matters in development for migration of the cells and disease in periventricular heterotopia. And we have, of course, many more candidates from this overlap. Now, I would like to continue these themes on two more organelles. One I alluded to already, the nucleoli, and we got into the nucleoli by this protein, TRNP1. In fact, it was cloned first here in Israel by Uri Nir in mammary epithelial cells, and it promotes self-renewal of these neural stem cells. This is indeed a nuclear protein. You can see it here. It has intrinsic disordered regions and undergoes phase transition in little droplets in the nucleus. And it has fascinating roles because indeed Its levels can lead to tangential expansion of a region because they amplify the neural stem cells. And when its levels are decreased in a block, it can elicit folding of the normally smooth mouse brain, because then suddenly you generate a lot of transit amplifying progenitors. Fascinating function, but how is it achieved molecularly? It took us a good 7 years to figure out only part of it. And what we found is that it interacts by its intrinsic disordered domains with several protein hubs in the nucleus. But in fact, one of these hubs is the nucleolus, and we focused on its role there and could show that indeed it promotes proliferation and translation by these specific interactions at the nucleolus. So without going much more into detail, this is also published, I'd just like to show you that at least for the starting protein, like with ACNA, in that case for TRNB1, we have shown that the nucleoli composition and function differs between self-renewing and differentiating neural stem cells, so closely related cell types, differ in their nucleolus composition by at least this single protein. So again, a common organelle seems to play a role there. So now I've been alerting you to this principle of regulation in development, but can we really use it to generate new neurons after injury? So after all this development, what does it bring us for making new neurons in an injured brain? Now, for that, let's move to direct neuronal reprogramming. And in fact, the start, when we started this more than 20 years ago, was of course in culture. And what we did was simply culturing astrocytes from the postnatal cortex and sticking in at that time the transcription factor PAC6 with which we were working. And we have further developed this to get better neurons by using pro-neural transcription factors, like well known for the developmental experts, but the names for the non-developmental crowd don't matter. With this transcription factor, we can generate glutamatergic neurons from these postnatal murine astrocytes, and with either of these transcription factor inhibitory GABAergic neurons, and they are functional and they form synapses. Now, the last protein I spoke to you is TRIMP1. It's important in neurogenesis, as I told you. It's actually also expressed in young postmitotic neurons, and we think it has it has another life there. But when we knock it down, we get a decrease in neuronal conversion from these astrocytes, and when we overexpress it, we get a doubling of the proportion of neurons that we get from these astrocytes from 30 to 50%. And its intrinsic disorder domain is essential for that. So we would conclude that its nucleolar role in the nucleus also plays a role here. Now let me close this organellar heterogeneity by showing you a key role of cell type specificity of mitochondria in direct neuronal reprogramming. And I'd like to just again summarize some aspects of this, but it brings us to the important aspects of in vivo direct neuronal reprogramming, and this is what I would then like to conclude with, because if we want to replace neurons in an injured brain, we need to do that, of course, in vivo. So, when we convert all these cells, either glia or, as Mario Zwernig has shown, fibroblasts into neurons, we realize that in fact there is a lot of cell death occurring, and in particular the young neurons, they actually die by a mode of death that is called ferroptosis, and that is due to excessive reactive oxygen species. Now, you may think it may not be so important to worry about it, but I would like to convince you how massive this effect is in vivo reprogramming. And this allows me also to introduce you to our readout for in vivo reprogramming. So we perform a dramatic brain injury, a little stab wound. 3 days later, we, in the adult mammalian moral cortex, 3 days later, we inject a Maloney-based retroviral vector that targets only proliferating reactive glial cells. And when we express just a fluorescent protein, not much happens. We don't get any young neurons, or old neurons. There is no endogenous neurogenesis. But then again, after starting these first with PAC6 in 2005, and then probing various neurogenic factors, we got some neurons, but they are clearly very young, immature neurons, even neuroblasts, nothing like a pyramidal cell that many of you know here, and you can look at them daily when you come to the institute. Clearly, these are not pyramidal neurons. So, for 10 years, again, we tried to find out what goes wrong, and this brings us to the ferroptosis, because when we found that, we then simply co-expressed with a single neurogenic factor, neurogenin 2, that normally just gives us 10% of these rather immature neurons. We co-expressed the factor that rescues them from cell death, plus treated them with antioxidants, vitamin E. And then you see that we go from these few percent to 80 to 90%. And not sick neurons, but neurons that start to look like pyramidal cells, have more mature markers, and much more we've also recorded from them. So, a massive improvement in direct reprogramming in vivo in this stab wound injury paradigm. Now, how does that relate to mitochondria now? I told you these guys die by ferroptosis. Normally, it interferes with their conversion, but why? Where does this ROS, reactive oxygen species excess come from? Well, maybe the mitochondria are not converted normally. And maybe mitochondria differ between neurons and glia cells. So when you turn a glia into a neuron, you better also convert the mitochondria from a glial type into a neuronal type. And that's exactly what we found. So first, we simply protein profiled, astrocytes, that case again in our in vitro system, and neurons. We found that they differ by 1/5 of their proteome, and of course the proteome also fits to some expected and known metabolic function that differ between neurons and glia. But most important for the reprogramming, we looked whether these neuron-enriched mitochondrial proteins are actually changing in the reprogramming, and we found that they don't. That none of these is actually upregulated by 2 to 3 days into reprogramming, and this is when the death occurs in this paradigm. So can we do something about it? We could, by using CRISPR activation tools to activate specifically one or the other of these neuron-enriched mitochondrial proteins. And as you see, some don't have any effect, and here we choose a low efficiency paradigm. Neither astrocyte-enriched mitochondrial protein activation had an effect, but others did have an effect, like SOD1 or SOD1 in combination with PRDX2. So indeed, it can help reprogramming when you activate even single neuron-specific mitochondrial proteins. And this shows you that also in this reprogramming paradigm, the cell type specific composition of mitochondria matters. And we've worked out the whole mechanism of why it is deleterious actually not to have the right neuron-enriched mitochondrial proteins because it mounts an unfolded protein response and actually slows translation. But I'm sparing you these further details for now. But what I would like to do in the last part of the talk is to dwell on neuronal reprogramming in vivo. Where are we? How good is it? Can we use it in the future? And then I think this will bring us naturally to a good discussion, hopefully. So I showed you already some inkling of that we can make 80% of neurons that still look a bit scrambled, but already are functional and have pyramidal cell morphology. But we really wouldn't want to use this in the brain of patients because I also told you we use the retroviral vector that targets proliferating cells. And this is how it looks when you look at the glial cells and the immune cells when you use such a virus in combination with this stab wound injury. So let's just simply summarize a huge reaction. There is other viruses that have a lower reaction, like the monocular virus or the AAV virus, the adeno-associated virus. So great, maybe now we can use the AAV to convert the neurons. And this is where some flaws in the field became apparent, and I'll show you how to overcome them, basically. So AAVs are in principle great tools because, again, when you think about therapy, you have some AAVs that you can actually inject IV, and they pass the blood-brain barrier, and they can even be cell type specific. So that could be fantastic. So we tested some of these that are claimed to be glia specific. Matteo injected them in the tail vein of mice and looked at simply 4 weeks later and finds indeed exclusively astrocytes labeled, very few neurons labeled. So highly selective targeting. We could use for reprogramming if there were not this specificity is lost when we perform an injury, actually. When we perform an injury, we get about 50% of neurons targeted. So these vectors are there, but they are not yet cell type specific. And of course, we and others are working on making them cell type specific. Why am I telling you this? Because this is the reason why several labs, including ours, then use a genetic trick to target the expression of our reprogramming constructs to the glial cells. But this is also where danger was looming. So what we and others use is this AAV 2.5 that definitely infects many neurons, but we make it visible, so to say, only in the glial cells or expressed only in the glial cells by using flex constructs. So these constructs are only inverted in the cells that express the 3 recombinase. When we do that with GFP, you can see that in this mouse line, we get virtually only astrocytes labeled. When we then put in our reprogramming factors, we have then replaced BCL2 with this other antioxidant transcription factor. We get not only many neurons, but also rather perfectly looking pyramidal neurons. Now this can be good news or bad news. It's good news because we want to make good pyramidal neurons, but it's bad news if these were artifacts as suggested by a paper from Jun Li-Sang Labs, who showed that when you direct the neurogenic 2 expression by the GFAP promoter in the same viral vector, you can actually get cis regulation in endogenous neurons. So, therefore, already prior to this paper, I was anyway very critical when I saw these perfect neurons. We performed many controls, and I just want to share with you one most important control, namely, we simply labeled the pre-existing neurons. And so far, we are still the only lab who does this control in direct reprogramming in vivo. So we simply label the endogenous neurons during development when they are generated by injecting EDU, a DNA-based analogon, being incorporated then. And then you see that many of the neurons are indeed labeled, the endogenous neurons here shown in red. 80% of them we can label. But then when in fact we look at the, here the reprogrammed neurons, here are the endogenous neurons, here are the reprogrammed neurons. When we look at the reprogrammed neurons, we see that they are, many of them are not EDU positive. So in fact, only 20% were EDU positive, i.e. endogenous neurons. So what I'm telling you that most of our reprogrammed neurons are indeed reprogrammed and not endogenous neurons. Now, it's never the end of controls, and so the best evidence for real conversion of the cells is in fact to image them live. And here you can see an astrocyte in live imaging through a chronic window, and this astrocyte actually over time turned into this pyramidal neuron. But of course we also found many neurons just appearing. So as also our fate mapping showed, we have both. We have some endogenous neurons that express the construct, it's a minority, and a majority of converting neurons. And this was also confirmed by further live imaging that I think I'll skip through. So what I've been telling you then is that yes, we can reprogram cells reliably and with good controls in vivo. It's of course cleanest if you use vectors integrating only in proliferating cells. Neurons don't proliferate, no problem. But these cause inflammation, and you don't want to use them in patients, so we need to improve the system. And AAVs, or the monocular viruses, are better in that, but they can also possibly target post-mitotic neurons. And you need to control for this, but anyway, I showed you there is clear evidence also with these factors to generate neurons. The last part I'd like to discuss with you, if I have still time, is that the next question, so to say. If using these beta viral vectors, we target also endogenous neurons, and we will do so in human patients, is this good or bad? What's actually going on? So, to look at that, we perform patch-sec to ask this question of the reprogrammed neurons, asking how good are actually the reprogrammed neurons in comparison to the endogenous neurons, and what actually happens to endogenous neurons expressing the neurogenic fate determinants, because that's what happens with the AAVs if we use them. So let me maybe skip through all the we use in vitro reprogramming of these factors. Here we use the NeuroD1, NeuroD4 because we find it more efficient, especially in combination. Then in vivo, we use two different viral vectors just to make sure, a monocular viral vector or the AAV. But let me now focus on the AAV. There again, using this combination of NeuroD1, NeuroD4, we see 80% of neurons, and we see only 10 to 20% in the control. These are functional neurons, and they are functional pyramidal neurons because from recordings, they share the parameters of electrophysiology with pyramidal neurons and not with non-pyramidal neurons. Now, the most important data I'd like to share with you is then the patch-sec data with, from the starter cells, the astrocytes, they are here in squares, and the converting cells, these are all the dots here, and the endogenous neurons. And again, this is a super important comparison because we need to know how good they are. And we did a very important control, as I think, namely we also activated neuroD1, neuroD4 using chem kinase 3 in endogenous neurons. These are in fact the green ones. Now, as you can see, when we plot after sequencing the data from all these 84 cells, we find the astrocytes on this part of the PCA plot. We find the reprogramming neurons, so again using GFAP3 to activate our reprogramming construct. And we took the cells after 2 weeks. Some are still with astrocytes, some are clustering with the neurons. And here you see the widespread of the various triangles, the endogenous neurons. Now, when you look at the endogenous neurons, what you can see is the bright dots. They are the intrinsic endogenous neurons without injury. The dark blue are the ones with injury. And the green dots are the ones that force express neuroD1, neuroD4. Now, from this analysis, obviously, we can do various analyses on looking at the conversion of the cells and the similarity to the neurons, as I alluded to. Now, what we can see here is just the same as I showed you already. The early reprogramming cells partially cluster with the astrocyte and partially cluster with neurons. We can also show that, in fact, the early reprogramming neurons still have high expression of some astrocyte genes, and that even the ones that cluster already with the neurons still have remnant expression of astrocyte genes. We take this as evidence for molecular conversion from astrocytes, because if we would activate gene expression in neurons, we wouldn't see that. I'm happy to talk more about that, but I'd like to conclude. So let's look at the comparison to the neurons now. Now here the black are the astrocytes, but here you see two clusters, if you want, of neurons. You can see the clusters of neurons. Here, the green ones, they are the ones that express neuroD1, neuroD4. And here are a few of the green, but mostly endogenous neurons. And you can see that the 4 weeks neurons, reprogrammed neurons, actually, again, cluster in those regions. But in fact, most of them are in this field. And this was also shown by the cluster diagram before. So, yeah, I think we don't need that. Let me summarize here then that, maybe still explain that. So this means that not all reprogrammed neurons that we collected after 4 weeks are similar to the endogenous neurons where we forced the expression, but rather many of the reprogrammed neurons actually are similar to the endogenous neurons. Now, interestingly, what you can see is that the endogenous neurons that actually express, I think I'll just summarize this, that actually express neuroD1, neuroD4, they become more similar to cells that have not been exposed to injury, but in fact are in the intact cortex. So, what I've been showing you with this last set of data is that the patch tag confirms the molecular transition of astrocytes to neurons, that some induced neurons are well comparable with the endogenous neurons, and that the neurons that we force to express, neuroD1, neuroD4, they actually seem to resemble more the neurons from intact brain. So it's not really probably a deleterious response, but we need, of course, physiology to work on that more. So with this, I hope I could convince you that there is great progress in the field of direct reprogramming, that we can convert the cells with high efficiency, But of course, we need to learn much more about network integration and the exact comparison at physiological level also to the endogenous neurons. And I've just started to show that to you. Now, can we do the same for human astrocytes? Now again, we probe this first in vitro, probing various factors to convert human iPS-derived astrocytes into neurons. And I will not bore you with the factors, but just say that this one, a mutant of neurogenic 2 was most efficient. And now, and this is I think where the future is now we can do that in vivo. By simply transplanting these transfused astrocytes that we derive from the iPS cells, either transfused only with GFP or with the mutant form of neurogenin 2, activate the reprogramming factor after transplantation, and as you can see, we lose some cells again here, but the ones we have, they turn into neurons after 2 weeks with high efficiency, and also after 4 weeks. Some of these examples here, and of course they need to develop much longer, and we can even see them live by a chronic window how they look. So, with this, I hope I could convince you of 2 things. One is that it may be important to look where a protein is localized in a cell, and that its function can indeed change, and it matters for disease where it is localized. And I hope I could convince you that cell reprogramming is maybe a cell replacement therapy 2.0. Much more work is needed, but let me just zoom out in closing and remind you that there is many diseases where functional cells are lost, not only after stroke, but also after a heart attack or in lung fibrosis. The scar cells take over, and converting scar cells into functional cells is an approach that I believe will be very important in the future. And with this zooming out, let me thank the fantastic people I have the privilege to work in the lab, our fantastic collaborators, funders, and you for your extended attention. Thank you.
Okay, time for a few questions. Students first. Tom.
I am a student.
Hi. I have a general question about the first part of the talk, about the changes in the composition of centrosome, nucleoli, and mitochondria in neural stem cells and mature neurons. I wonder whether it's true also outside the brain, that the progenitors have different compositions compared to the mature cells?
Yep.
And if so, whether they can be potential targets for preventing the opposite effect of excessive unregulated growth like in cancer.
Mm-hmm. Yes, very, very interesting point. So yes, indeed, on the level of individual proteins, it's known that, for example, also in skin differentiation, the centrosome proteins, some centrosome proteins change. Often in differentiation, the centrosome actually loses this activity to nucleate and organize the microtubules. That has also been shown in skin and muscle cells, but there is no comprehensive proteome. Now, to use centrosome proteins to manipulate proliferation is a cool idea. It could be that the proliferating factors would still override it, and you would then get even worse a situation if you have even more abnormal proliferation. But definitely manipulating centrosome proteins and or bringing them to the centrosome where they may be normally there and are not in certain diseases is something to also consider and use for therapeutic manipulation. So manipulations to help, absolutely.
you are doing this in vivo and you're reprogramming astrocytes, what happens to the rest of the cells there? They're lacking astrocytes. I mean, small numbers, but still they're missing cells. So as they're like in proliferation of other astrocytes reduced, or how do they compensate?
Exactly. So as we already spoke during lunch, actually in most neurodegenerative diseases, astrocytes don't proliferate. They proliferate only after invasive injury, and only few proliferate. But in this traumatic brain injury, they do proliferate. Now, we are far from converting all of the astrocytes and all of the proliferating astrocytes, but to tackle the question, what is the effect on the endogenous neurons, we actually performed these patch sac experiments to see if they would actually change in a deleterious manner, but in fact, this doesn't look like it. But rather, we see also for the endogenous neurons in the vicinity of the reprogrammed astrocytes, we see molecular changes that go into the direction to the intact neurons. But it's early days. And of course, gene expression from patch-sac is of course just a start. Let me just emphasize that. So certainly we need to understand the effect on the surrounding, but let me again just emphasize, we don't know this in transplantation, and transplantation is already used in the clinics, right? We are doing the same, studying the surrounding after we transplant neurons, because we are always comparing, but I spared you the transplantation part. But so, very important principle. We may not have to solve it before using it in patients, at least for transplantation, this is not needed.
Last question. Chaya, for the sake of time, we'll take one more.
Okay, if you touched upon the idea of transplantation versus direct endogenous reprogramming, could you elaborate a little bit more on the efficiency, on the relative efficiency of each one of the protocols in terms of eliciting neurogenesis, and in particular in terms of affecting maybe neighboring cells, because there are cell-cell adhesions and all kinds of interactions about which you didn't talk at all. So could you elaborate a little bit on that?
Yeah, I mean, so the second part, as I just answered, nothing is yet known, and we are starting to look, but the transplantation field also very little is known on how the transplanted cells actually affect the environment. There is a recent paper on transcriptome analysis, and in fact, again, it seems that the transplanted cells rather decrease the reactivity of the glial cells in the environment. This is a paper from Mike Sofroniev. But other than that, population analysis, relatively little is known. But we need to study it. But again, comparing transplantation and reprogramming is something that indeed we do in the lab, because for us, the transplantation is always the gold standard. And looking at the gold standard, we find still some basic issues to be solved. For example, the transplanted pyramidal neurons, they are not myelinated. Nobody has seen that so far. Nobody had looked. So, you know, the moment you look, you see what is not yet working perfectly. If you don't look, you don't see it. So I think both approaches are very important. Transplantation has started 20 years earlier. It's closer to the clinic. The direct reprogramming has the advantage that it could work for targeting widespread regions using these systemic AAV injections, while transplantation would typically be better for focal diseases where you would inject your transplanted cells. In terms of efficiency, I think the reprogramming can be as efficient in terms of numbers in murine mouse models. Obviously, we haven't done anything in patients yet.
Let's thank Magdalena.
you.